So water's been shown to retain information. And we've learned this through the multiple decades of research with homeopathy, for example, and all of the clinical effects that we've seen with homeopathy. And Dr. Jacques Benveniste, a French scientist, was one of the first people to design an experiment that was published in a reputable journal, actually the journal Nature. And his study was very controversial. But what he showed was that he took IgE antibodies and he put them in water and then diluted the water down. So he took the water that had the IgE antibodies in it and shook it vigorously, and then took some of that water, put it in a vial of pure water, shook that vigorously, and kept diluting it down until there were no IgE antibodies left in the sample. Now, IgE antibodies are what produce allergic reactions in our bodies. They actually trigger cells called mast cells to release histamine. And what Dr. Benveniste was able to show was that the water imprinted with IgE antibodies were able to cause mast cells to release histamine just like IgE molecules did. And like I said, this was very controversial and kind of made a storm in the scientific community. Dr. Luc Montagnier, another French scientist who was a Nobel Prize winning scientist before the discovery of HIV, was able to pick up where Jacques Benveniste left off and even work with his same scientific team and do further research on this topic. And what Dr. Montagnier did is he took DNA from an HIV infected patient and put it in water and diluted the water down and was able to use a computer to measure vibratory information from molecules. And he was able to show that even after the water was diluted to the point that there were no molecules left in the sample, the computer could still read the information vibratory signature of the DNA. And then he took his research a big step further and collaborated with researchers in Italy. And he took this now computer digitized signature of the HIV infected patient's DNA, and he transferred it to a lab in Italy. And then the lab played the signature to water. And what Dr. Montagnier said is that the water listened to the information. And then they were able to show that water that had listened to the information still retained that information and compared that to pure water that had not listened to the information. And then they took it a step further and they added single nucleotides, which are the building blocks of DNA, and the enzymes required to assemble DNA into water that had listened to the signature and water that had not. The water that had listened to the information was able to reassemble the DNA. So it's almost like you buy a piece of furniture and you need the directions in order to assemble it. So that water had those directions, and the pure water that had not listened to the information could not. And they took that DNA that was assembled by the water that had listened to the information, sent it to an independent laboratory that did PCR analysis, and found the DNA was 98% similar to the DNA in France. This was extraordinary on so many levels and took the scientific community by storm. It's interesting because people are still very close-minded about this, but to me, this is absolutely undeniable. And so how is it possible that water can hold information? It turns out water has little pockets for holding data. So water molecules will arrange in such a way, almost like children holding hands. So you have oxygen with hydrogen atoms on both sides, but now you have a ring of these where oxygens are sharing their hydrogens. And this ring sets up what's called a coherent domain. And the coherent domain excludes other types of molecules, such as ions or sodium or chloride or things that might be in the water. It's a pure coherent domain. This area has been shown to be a perfect pocket for holding data.